Good morning. So for my journal club, I'll be discussing this article, benefits of clinical criteria and high-throughput sequencing for diagnosing children and syndromic craniosynostosis. So this article was published in December 2020 in the European Journal of Human Genetics, impact factor 4.9. The study setting was in the Oslo University Hospital, which is situated in Norway. So following terminologies we should know: craniosynostosis, it is early fusion of one or more skull sutures in infants leading to abnormal skull shape, restricted brain growth. It can be either syndromic or non-syndromic. Then high throughput sequencing, it is a rapid DNA sequencing technology that allows testing of hundreds of thousands of genes at once. Exosome sequencing is high throughput sequencing only of protein coding genes, where mostly the disease occurs. The trio-based sequencing, it is genetic testing of child and along with both parents to detect new de novo mutations and inherited variants. The custom designed gene panel, it is a set of selected genes known to be linked with particular disease. In this case, this study, 72 genes were known for syndromic craniosynostosis. The extended gene panel, it is a large group of genes tested to detect rare, unexpected syndromes beyond the common ones. Single gene analysis, it is testing of only one gene at a time, usually when specific syndrome is suspected. Array comparative genomic hybridization, ACGH, it is a test used to find large deletions or duplications in chromosomes that may cause disease. The CHART syndrome is one which is discussed here. It is an autosomal dominant disorder due to CHD7 gene mutation. The CHART is an acronym for cranial nerve abnormalities or cobolemia, that is hole in the eye, and microphthalmia, heart defects, atresia concae, that is blocked nasal passage, growth which might be restricted or delayed, and ear anomalies and hearing loss. Chromatinopathy, it is a disease caused by mutation in genes that regulate DNA packaging and gene expression, that is in the chromatin. For example, in Cliff-Strauss syndrome. Mosaicism, it is a condition where a person has two or more genetically different cell population in their body due to mutation occurring during the fertilization stage. The de novo mutation, it is a genetic change that appears first time in the child but is not present in either parents. Deciphering developmental disorders, it is large gene database used to identify gene associated with developmental problems. FGFR genes, Twist1 genes, and EFNB1 genes, these are genes that mainly control the growth of face and cranium, and mutations will cause various problems. Common symptom for these is craniosynostosis. The ClinVar, it is a public database where genetic variants and their disease associations are reported. So this study, novelty, unlike the previous study that focused only on a few known genes or physical skull patterns, this study combines a defined clinical criteria with a stepwise genetic testing approach, including high throughput sequencing of 1570 genes. This is one of the first large population-based study to show such a high diagnostic success rate for syndromic craniosynostosis. So the authors, they hypothesized that using broad genetic testing combined with detailed clinical criteria would improve the ability to detect and confirm the genetic cause in children with craniosynostosis. They aim to evaluate how and define how well the criteria and the advanced throughput sequencing can identify the underlying genetic cause of syndromic craniosynostosis, where their objectives were to classify children with craniosynostosis, then apply stepwise genetic testing strategies, and to determine the proportion, that is diagnostic yield, that can be genetically confirmed, and to describe the range and diversity of genetic syndromes found in these children. So the study design, it is a population-based study. The data from Norwegian National Unit for Craniofacial Surgery was taken from 2002 to 2019, and all children were suspected craniosynostosis, were evaluated clinically and genetically. Ethical approval was taken, and the participants were 381 children diagnosed with craniosynostosis were included, out of which 104 were classified as syndromic based on the clinical criteria. The clinical classification were done where if the children had one major criteria or two or more minor criteria. Major criteria are severe developmental delay, autism, organ defect, severe facial malformation, or minor criteria are mild developmental delay, vision, hearing loss, hearing, mild dysmorphism, growth abnormalities. So they standardized the diagnosis before testing the genetic testing. So the genetic testing, they did single gene testing for known craniosynostosis genes. After which they did the array comparative genomic hybridization to detect a large chromosomal anomalies, followed by custom designed high throughput sequencing panel. It sequenced for 72 known craniosynostosis related genes, which if earlier which was negative, they were tested through high throughput sequencing, and extended trio based HTS panel where they sequencing of 1,570 genes from the database of developmental disorders for unresolved cases were done. The sample processing, they collected blood samples for DNA extraction. Sequencing was done by using Illumina high sequence system, and the variants were filtered by frequency and pathogenicity using these softwares, and only pathogenic or likely pathogenic variants were reported. The data recording, the results from each test were added to national registry and compared across methods to measure diagnostic yield. The statistical analysis, they measured the diagnostic yield basically in percentage, and comparing the success rate between different genetic testing methods, which served as key analytical outcomes. So the results, the cohort findings were out of 381, 104 had syndromic craniosynostosis, that is around 27%. Out of these, 104, 94 agreed for genetic testing, and thus diagnostic success 73% identified by single gene testing, ACGH and HTS, whereas this rate increased to 84% after extended trio-based sequencing. The genetic causes, 56% of the participants had a mutation in the common genes, that is FGFR2, twist1, the EFNB1, and whereas the 28% had rare genetic syndromes, that is 23 different ones, and 16% had no genetic diagnosis. The key syndromes which were found, the common one were Apert syndrome, Munich syndrome, Satter-Schwartz syndrome, Crouzon syndrome, craniofacial dysplasia. The rare syndromes that were found were Charge, Kliefeld, Brainbridge, Roper's syndrome, Malin alpha mannosidosis syndrome, Floater Harbor, and others. So there were various syndromes which were found, and the chromosomal anomalies in ACGH results. The 10 children had deletion or duplication, which some were linked to craniosynostosis. Then custom designed HTS panel results. So they found that mutation explaining both typical and unexpected presentation, including mosaicism. and new genotype phenotype links were found. The extended trio-based HTS panel. So this revealed that very rare syndromes not classically tied to craniosynostosis, showing how broad genetic testing uncovers the hidden causes. The novel observation that was seen was they detected mosaicism in some parents, explaining the recurrence in siblings. They found chromatinopathies, that is disorders of gene controlling DNA packaging like Charge and Craniofacial syndrome, suggesting also these can cause craniosynostosis, and identified cases with single midline sutures rather than multiple fusions, indicating different molecular pathways. So for discussion, the combination of clinical criteria and broad genetic testing enabled high accuracy identification of syndromic cases. The study confirmed major known syndromes but also expanded the list to include rare and ultra-rare conditions. It also showed the role of chromatin modification disorders causing craniosynostosis, which was a new finding, and the authors emphasized the importance of multidisciplinary teams along with long-term follow-up for early detection. They also recommended using stepwise testing for cost-effectiveness and accuracy. The strength of this study, it was a large population-based prospective study for over 18 years, and they standardized clinical assessment and genetic testing for all cases. One of the highest reported diagnostic yield for syndromic craniosynostosis was seen, that is 84%, and inclusion and of rare and mosaic genetic findings were also included. So these are the strengths. Limitations, some recent craniosynostosis cases may not have developed additional syndromic features yet, that is underestimation, and some genes such as these were not included in the older test panels. A few patients did not undergo extended HTS testing, and potential for missing deep intronic or regulated mutations not detectable by the exosome sequencing. So these are limitations. Future scope can be incorporation of newly discovered genes in the testing panels, and they can study the molecular mechanism behind chromatinopathies and skull leading to skull fusion, and they can expand international registries to compare genetic patterns across population and the whole genome sequencing to capture undetected or non-coding mutations, and to establish standardized clinical definitions globally for better research comparability. So the study design, the study was a prospective population-based registry model. So this is well suited for rare congenital anomalies. The overall compliance was high, where they met 22 out of 30 criteria fully, and 8 out of 30 criteria were partially met. The partially met criteria were near completion inclusion of eligible cases, that is 85% of the eligible patients were consented, small percentage were untested, prospective follow-up where the data collected longitudinally but long-term clinical follow-up not detailed beyond diagnostic completion. Then comes the detailed description of data source, where the registry source was described but specific data variables were not listed, and followed by data quality control, where bioinformatics, filtering and variant validation was explained, but registry data verification not explicitly discussed. Incompleteness of data reporting, all 381 CS cases were described, 94 syndromic testing, but small subset was untested, and the handling and listing of incomplete data. The non-participants noted but formal missing data strategy not stated. Then comes long-term sustainability. The registry updated to 2020, but ongoing maintenance were not discussed. And comparison with other populations, we briefly compared diagnostic yields with other studies, not in depth. So these are the criteria which were partially met. Thank you.